This is the second lecture for Comparative Genomics. Today I'm going to be talking about evolution and genetic change. And hopefully by the end of the lecture, students will have a better understanding of the coordination of changes in phenotype and genotype during evolution. Also, an appreciation for the distinction between similarity and homology. They'll be able to recognize the measurements of similarity between genes and protein sequences, and that offer insight into evolutionary relationships between individuals and species. They'll understand the concept of pattern recognition and the basis of phylogenetic tree construction. So in other words, as we talk about evolution and genetic change, we're going to be talking about sort of, what does it mean to put patterns of similarity and differences into an evolutionary context, and why is that important? So again, the name of this class is Comparative Genomics, and anytime you hear the term comparative, you should automatically think that has something to do with evolution, because what we're doing when we do comparative work is we're using different biological systems to understand some sort of process or trait or mechanism, right? And it's only within the context of the history of that organism or that group of organisms that we really are able to understand how genetic change has occurred and how that genetic change has led to some sort of phenotypic change. And that is basically looking at how the effect of evolution. So it's a really important component of understanding how evolutionary change works, how genetic change works, so we can better actually understand function. So I think a really important part here is how are we describing evolution? I think a lot of people think of evolution we hear it in sort of popular culture. It can have a lot of political charge to it, and people often misunderstand it. Evolution is actually, a field of biology that is actually pretty hard to understand, but I think it's underappreciated just how mathematical this field is. And, to an evolutionary biologist, what we're really looking at is a very explicit way of tracking genetic change through time. And how we do that, and in sort of like the most basic definition of what evolution is in looking at changes in allele frequency over time, right? Doesn't sound all that exciting, but it's really this heritable genetic change, this change that is occurring through time, that really underlies and legitimizes the process by which we conduct evolutionary investigation. It's important we define then, what do we talk about when we say allele, right? So an allele is a sequence variant that occurs at a particular locus. So we're seeing allele frequency changes over time. We're saying if I look at a group of organisms and I'm seeing the sequence variants change from one type of variant to another type of variant, that frequency, that representation in the population, then that's evolutionary change. For that to occur, I'm going to have a polymorphism in my population. In other words, I'm going to have allelic variation at a locus that is greater than one, that is found in greater than 1% of the population. So I can't have evolution unless I have variation. It's a very important point. And when we think about, okay, so I need to look at changes in allele frequency over time. For there to be changes in allele frequency, I need variation. So what are the different things that can affect patterns of variation? And there's basically 5 main evolutionary forces. And again, these forces are the things that are changing allele frequencies through time. So things like drift and mutation right? Drift is the random sampling of genes from a population. It's considered to be a neutral process. Mutation is also considered to be a neutral process, right? And mutation is one of the only ways that we ever get sort of like the generation of new genetic material. There's migration, in other words, the movement of alleles between populations, right? It's another way to change allele frequencies. Recombination, the crossing over of genes, and then finally selection. And this is one of the big ones we always talk when we talk about evolutionary forces, is how selection is driving evolution. And I think it's important to know that selection is one of 5 here, right? So it's the one we talk about the most when we talk about evolution. But, all these other 4 evolutionary forces also can cause evolution to happen. And it's important that we recognize that. I think when we think about evolution, especially from a comparative framework, and it's a little bit different than how we might think about in other frameworks, because we're taking more of a macroevolutionary perspective. But we're really interested in what are things that are similar and what are things that are different. So, we want to show change over time. So to do that, we have to look for these patterns of similarity and difference. Historically, when we have done this, we looked at anatomy, we looked at embryology, we looked at paleontology, right? We looked at all these different samples, all these extant species, all these or fossil record, and we tried to say, okay, well, given these patterns of similarity and difference, what is the most parsimonious, or what is the most likely explanation for the relationship among these things? So we could look at a group of organisms, say like these cichlids over here on the right side of the screen. Right, and these are cichlids from different lakes in Africa. And if we were to look at this species, phenotypically, there's a lot of similarity between these species in these different lakes. And we might think that, the most closely related ones are the ones that are actually phenotypically or physically similar to each other, whether we're going by body shape or coloring or feeding adaptations, whatever we might see there, right? But it's when we look at genetics that we start to really sort of be able to get a better sense of what are the actual similarities and differences, and what are the components that are underlying these things, right? So we also get a much clearer picture of what is the history of these things. So when we look at something, are things that look physically the same, similar to what we would see based upon their genetics? Are they the most closely related? And this is a classic case, looking at these cichlid speciation events and speciation patterns. And if we look at these organisms, what we find is that one group is from Lake Tanganyika and the other one is from Lake Malawi. And interesting, what we see is that the genetics tell us a very different story than just what we might get from looking at them phenotypically. So if we look at them genetically, what we see is, if we look at the phylogenetic tree, or the tree explaining how these groups break out genetically, we can see that the Lake Malawi fish are all more closely related to each other than they are to any of the fishes that are found in Lake Tanganyika. And if we look at how this works out geographically, we can see Malawi's down here, Tanganyika's up here. It looks like, the fish of Malawi and Victoria here are more closely related to each other, so they all come from a common ancestor, and then diversified into phenotypic groups or classes of groups that look very similar to the ones in Tanganyika. Right, but they are more closely related to each other than they are to fish that we find in Tanganyika. And so what this indicates for us, right, is that we have this case where phenotypically they look much different. We might make a much different conclusion than what we see from them genetically. How do we build that kind of tree? How do we see these relationships between these groups? Well, what we're doing in that case is we're actually looking at patterns of genetic similarity. So what we're doing is we're classifying these groups of organisms or these clades of organisms, these branches, these groups of branching parts of this tree, and we're clustering them together, right, based upon their genetic similarity. And you can see here is an example of a multiple sequence alignment. So in other words, it's a sequence alignment. It's the DNA sequence at a specific location or locus in the genome. And we're aligning these and we're seeing, that there are groups of organisms that have similar genotypes. And these genotypes help define these different clades. So we can see up here, right, there are specific combinations of mutations that we associate with a group of organisms, right? And based upon this, we'll start to cluster out and form these clades, these groups of organisms that have sets of adaptations that are all going to go together. Right, and this clade, for instance, is defined by this large indel, right, and these other series of sequences. So that brings us to sort of, one of the big concepts when we think about evolution, which is the species tree or the evolutionary relationships, the phylogeny of a group of organisms. It doesn't always have to be at a species level. It could be at a population level. It could be among individuals. But usually when we're talking about a phylogeny, we're talking at it in terms of what is the evolutionary relationship between a group of organisms, typically at a species level, right? And so it's not always a species level, but it's often when we were talking about phylogeny is what we're talking about. And so one of the key principles here is that when we think about what is responsible for these branching patterns that we might observe, say if we're looking at flightless birds, is that things like cassowary and emu are more similar to each other than they are to kiwi genetically, right? So that would indicate that they have some sort of common ancestor. And that these three species, cassowary, emu, and kiwi, are more genetically similar to each other than they are to ostrich, right? And then this entire clade here is more similar to each other than they are to moa. And then this group is all more similar to each other than they are to rhea, right? And that's the idea about how we build this tree. Now, there's a lot more that goes into how these branches are actually drawn and how these divergences are often drawn. But the important part here is to show that is to understand that based upon this model of evolution, that we would predict that cassowary and emu would have a more recent common ancestor than the most recent common ancestor between cassowary, emu, and kiwi, which would be back here in time. So phylogenetic trees almost always, you have leaves down here at the bottom. They have the root up here at the top, which is the common ancestor. And you'll have various common ancestors, most recent common ancestors for each of these clades. So we're actually going back in time as we go up this tree. Okay. Phylogenetics is is a term referring to a type of phylogeny that uses an approach that uses a type of phylogeny that uses character data or observed traits to describe these evolutionary relationships. And when we talk about character data, historically it had been through things like, morphology, morphological characters, jaw size, things of that sort, right? And nowadays, this is more driven by using sequence information, because the sequence information, if we think of this like a family tree, right, is going to be much more indicative of the real genetic relatedness or genetic similarities between these groups. Those phenotypic characters, those morphological characters, as we saw with the case with the cichlids, can really obscure what might be the true patterns of relatedness among these organisms. So after we have drawn this entity right here, we refer to this as what is actually the phylogenetic tree, right? And this is actually what's portraying the evolutionary relationships. Sometimes these branch lengths are proportional to evolutionary time. So you will see that, the longer the branch length, the longer it's been since a convergence or a coalescent event has happened where these where you find this most common recent ancestor. So in other words, the most common recent ancestor between cassowary and emu was a lot more recent than the one between emu and moa, for instance. Where do we apply these trees, right? We use trees all the time in evolutionary biology. There are one of the dominant data analytical approaches or metaphors or visualization tools that we use to describe the relationship between species. So we can look at, this classic work by Carl Woese, and it shows us, the breakup that we see between sort of the major divisions on the tree of life with archaea, bacteria, and eukarya, right? Where we see that the point here is that, bacteria and archaea, right, are more similar to each other than they are to anything, and eukarya are more similar to each other. They form these groups. And one of the interesting things about this was that, archaea was actually more similar to eukarya than bacteria was similar to eukarya. So this would indicate, a closer evolutionary relationship with archaea as opposed to bacteria. Again, thinking about these patterns of genetic similarity, right? This is what's allowing these sequence similarities are what is responsible for the formation of those different branches that we just looked at. And the important thing to remember here is that similarity is basically genetic similarity. So similarity in the DNA sequence is really what is going to be accounting for this relationship among these different leaves or these different taxon, right? That's what we're looking at. Again, if you think about what is the dominant paradigm in comparative genomics, we're looking for things that are similar and looking for things that are different, right? I think it's important, too, that when we talk about these ideas of similarity and difference, you want to think about, okay, well, we found this locus where we're looking for similarities, but what we have to kind of be doing is looking at the same genetic position, locus, marker, gene, however you want to define it, in multiple different species, right? So if we're looking at speciation, we want to be comparing apples to apples and not apples to oranges. And so it's important that we do that we make sure that what we're looking for to identify are homologs that we find among these different organisms. So if I was to talk about a homolog, what is a homolog? A homolog is a gene that's related to other genes by evolutionary descent. Evolutionary descent from a common ancestor. And when we talk about homologs, there's actually generally two different types, major types, orthologs and paralogs. So an ortholog is what we're going to be using when we compare species. So this is one gene of a set of genes that descended from a single gene and a common ancestor. And these sets of genes diverge from each other due to speciation. So the example here is if I have gene A, right, and this is the ancestral gene, right, when there's a speciation event, what I end up with is species 1 having an A gene and species 2 having an A gene. Same thing with gene B, right? When there's a speciation event, I'm going to end up with species 1 having a copy of a B gene and species 2 having a copy of a B gene. And so I can then compare these things to each other. I'll compare A to A and B to B. Another type of homolog, which is really important to consider, especially in the field of comparative genomics, is when we're looking how well we can identify paralogs. So in paralogs, what we're talking about is one gene of a set of genes that underwent a duplication event in a common ancestor. And this is where the set of genes diverge from each other and then the sets of genes diverge from each one another due to the evolution of a new species, right? So in this case, we can think of an ancestral gene, right? And there's some sort of duplication event in that gene, and then we end up with an A copy and a B copy, right? Now, in the future, every species that evolves from this organism, species 1 and species 2, right, will have both an A and a B gene and an A and a B gene. Right, so species 1 has an A and B, species 2 has an A and B. So A and B are paralogs to each other. This can also happen downstream, right? Where like we could have an A gene, right? There could be a speciation event, and if duplication occurs afterwards, then we would see that both species would have an A gene, but since duplication occurred afterwards, there'd only be, in species 2, we'd only find that copy, that B gene. And this is important when we're, especially important when we're doing comparative work, because when we're doing comparative work, it's really important that we're comparing apples to apples and oranges to oranges. We're not comparing apples to oranges. So we're not comparing A1 to B2. We're comparing A1 to A2 and B1 to B2, not A2 to B1 or A1 to B2, because the way that these sequences evolve could be very different based upon whether or not it's an A gene or a B gene. Again, this is really important when we start to think about, okay, what are the factors that are actually going to explain and establish this tree? Because obviously in a case like the one that we talked about earlier with cichlids and their speciation models, it's really important that we're able to cluster out these things appropriately so that we can see whether or not we're looking at a pattern that's because of evolution that happened within the Lake Malawi or that happened across these. And this is a really interesting case where we see incredible what's known as convergent evolution, where these organisms are converging on the same phenotype even though they have different evolutionary histories. So a very interesting case. And it's also important to note that when we think about what is the data that went into this tree, right? What it is, it's actually multi-locus data. So we're looking at different genes and different organisms. So we want to make sure that we're always comparing the same orthologs in each of these species, and we're not worried about, we're not comparing paralogs from one organism to a different paralog in another organism. When we build that tree, though, one thing that you might wonder is like, well, how do I decide these things, how they diverge and what are these little numbers here? We can see that there's different lengths now. And down here we have a sort of a scale bar for the rate of evolutionary change. But what accounts for all these different components? How do I know that, what the length of a branch is, what are these numbers that I'm looking at, etc. Right? And these numbers right here actually indicate support for this clade all being accurate. So what these are basically referring to are what are known as bootstrap values. And these bootstrap values basically say, well, if I was to redraw this tree, using some random sampling methods, some statistical methods that cause, I can kind of like mix up the data a little bit. If I do that, 100% of the time, these two species always end up in the same clade, no matter what I do to the data. How much I might randomize it a little bit, right? And that 99% of the time, these three species all end up in the same clade. So what this does is it sort of establishes how much support you have for these different groupings of organisms across your tree. How do we establish these groupings, and why are we so obsessed with similarity? Well, it's the relationships between similarity that we think not only help explain this tree structure, which is so important to us, but they also have a relationship between function. And the argument here is that if we have sequences that have similar sequence, then they also might have similar structure, three-dimensional structure. So, as they form proteins and these proteins fold, if they have similar sequences, we'd expect them to look the same. And in addition to that, sometimes, what we actually see is that we can have structures that might be very similar to each other, but they don't have sequences that are all that similar to each other. And sometimes we have sequences that are very different in their sequence and very different in their structure, but actually might do the same things, right? And this just is one of the sort of challenges in protein biology and biology in general is understanding what is the relationship between sequence, structure, and function. We would, in the easiest case, things that are of similar sequence have similar structure, have similar function. But we know that that's not always the case. So how do we identify these patterns of similarity, right? One of the ways that we go through and start evaluating whether things are similar to each other is by doing something referred to as a sequence alignment. And this is not a bioinformatics class. I'm not going to spend an inordinate of time talking about the methodologies that we use, but I think we should have a basic understanding of what we're doing, because we're going to talk a lot about papers. We're going to want to think about what are the actual, how to interpret and understand the data that's being presented. So I'll talk a little bit about this stuff as we go through, but I'm not going to spend a lot of time going into the methods. If you really want to learn about the methods and how to apply them appropriately, you should take my bioinformatics class, which I offer in the spring. And if you're interested in that, just understand that there's a prerequisite that you would have to take the practical computing class that I also teach here. And the reason why is that we do a lot of work with computational tools, and so you need to know a little bit about command line and programming and stuff like that to be able to do the work of the class. That said, sequence alignments themselves are not all that difficult. You've probably done them at some point without even realizing that was what you were doing. But basically what we're doing in this case, right, as I showed you some examples earlier, we're using them to measure the similarity we see between sequences. And typically how they work is we take a series of sequences, a group of sequences, we do some pairwise comparisons, and then based upon the patterns of similarity, we'll then align those sequences to each other, right, accounting for any differences in the sequence. We'll then compare the patterns of conservation and variation, and based upon those patterns of conservation and variation, of similarity and difference, we'll then try to infer the evolutionary relationship or the homologous structure and function of a group of proteins. So how do you actually make a molecular phylogeny? How do you define it or reconstruct it? Basically select and align some sequences. You then will apply a mathematical model, the best model that most closely matches your data. That you'll try to model the evolution of these sequences. And once you've done that, we'll apply some statistical methods to find the tree with the best topology or ordering of organisms into clades and or branch lengths that describe the data. And then once we have the tree and the phylogeny, then we can actually interpret the resulting tree in the context of whatever question we're asking. It's important here to understand that there are these different, what do I mean when I talk about models of evolution? What I'm really talking about here is these kind of rate matrices, which just basically tell us, okay, if things were to change, if you're going to have an A evolve into be a G or a C or a T, what is the rate that we would expect that to happen? Are there certain things that are more likely, certain things that are less likely? And one of the things that we find is that, we could just define it where, if you have an A here, you're equally likely to evolve to be a G, a C, or a T. Or if you are a G, a C, or a T, you're just as likely to evolve to be an A as anything else, right? So it's all equal transition probabilities or mutational probabilities. There are, however, other cases where what we might see are differences between transitions and transversions, right? And so this is where, pyrimidine may be more likely to evolve or transition into being, or mutate into another pyrimidine, and a purine might be more likely to mutate into another purine. Right, and so this is what this example is, where we have different rates of these transitions versus transversions. We're going from a purine to a pyrimidine or a pyrimidine to a purine. And this is actually generally what we see, that there are real differences. And that could be due to the biochemical properties of the organisms, which mutations are likely tolerated or more likely to happen. But it is definitely the reality of the situation. We can also see There are other models like comparing where we see like an equilibrium in frequency, or we have all rates independent from each other, each of them created by different people to basically explain different patterns of evolution that could occur. So once we have that pattern of evolution, then we can use it to explain the relationship between, you can see which model best explains the relationship between a bunch of sequences that have been aligned to each other. So if we have, these two sequences, right now it's going to align them to each other. What we see is that the best alignment for these two species, or these two sequences, involves inserting an insertion deletion here and here, right, to align them, because now we have C, D, and E all aligned. We have A aligned. We just have differences at B and F. Same thing if I take a sequence like these two sequences, where I might know that there's a certain gap position at the beginning. I could try to align them. I could force the alignment just over the sequence. Doesn't look like a great alignment when I do that, but I do have some alignment here between these two A's. But in actuality, as I go to align them, maybe it looks something more like this. This is a better alignment of the two. Still not great. Like, I'll keep trying to do different types of alignment and try to find the most similarities between these two sequences. And so here's a case now, right, where I have the C and the T match up. I've got the A's matching up, and then the C here. Right, and so you can see how you can iterate through these processes and try and insert gaps and move these pieces around until you get the thing that looks the most like the thing that the most similar, that represents the patterns of similarity and differences the best, right? So we can make an interpretation of the loss of, or the insertion of, sequences at different parts of these multiple sequence alignments when we do this. One of the ways that we interpret this type of sequence alignment data is, especially in comparative genomics, is through a method or a conceptual visualization tool known as dot plots. So dot plots, here we have a case where we have the alignment between two strings, or two series of letters. So there's Dorothy, Crowfoot, Hodgkin, and then there's Dorothy, Hodgkin. And obviously, Crowfoot is missing from the second term. And that's represented here, right, where we have, all the matching letters on the diagonal, and then we have a gap here where we have nothing that matches in this species on the left, in this sequence on the left, and that's represented by this break, right? And then we have a continuation of matching there. We can also, when you have things that look like repeats, they can look like this, right, where we see them repeating each other, right, over and over again, and they end up with these parallel diagonal lines. We have cases where we could have an anagram, sorry, not an anagram, a reflection of that sequence. So, in this case, we have the same sequence, but reversed in both directions, and we can end up with this crossing location, right, or crossing motif. And so dot plots are generally applied in lots of cases, and if we had to think about what the actual multiple sequence alignment would look like in this case, it would look something like this, where we have a gap insertion for this type of alignment between two sequences. And so, like, this is an example of looking at the relationship in a specific gene, say ephrin B3 proteins from human and Xenopus, a frog. And if we look at these cases, right, we can see that we have regions of these two sequences that are very similar to each other, right? We have gaps where there might be a sequence which is missing from one of these organisms. You can see here, cases like this where like one species has something there and the other species doesn't. That would be indicative of something like this. We can see regions where we find stuff that's parallel to other parts of the genome. This could be regions that are similar to this part of the sequence or repeats. So, by understanding this, we can start to identify parts of the gene where we see conservation, right? Things that are conserved along the diagonal, and parts of the gene where we see differences, where they're either not on the diagonal or there is a gap or a lack of information in that section when we compare the two. One thing to consider that's really important when we talk about these dot plots is how we can use them to look at things like rearrangements. And this is a really important data analysis technique and data analysis outcome from comparative genomics, because we're looking at the whole genome. It's not like we're looking at just one gene. So by examining patterns of rearrangements, translocations, inversions, gene amplifications, right, we're now able to actually see how the whole genome is evolved. So the order and the placement of genes throughout the genome. And that can be really important. So referring to this basic field of this category of things as genomic rearrangements, right? We have things like the translocation events, which are mutations where one part of a chromosome fuses with another. We have inversion events, where a segment of a chromosome breaks off and inverts, but reattaches itself to the same chromosome. There are gene or DNA amplifications, duplications, where a segment of the genome will sometimes repeat itself or make another copy of itself. And so we have multiple copies, segments of what was the original ancestral genome sequence now in multiple places. And when we're doing all these comparisons for genome arrangements, it's important that what we have to use as a tool is look for regions of synteny, where we have similar arrangements of genes in the genome of two or more ancestors from a common species. So we're looking for the order of genes, regions that are similarly ordered when we're looking for patterns of synteny. Over on the left side shows an example where a genomic rearrangement, right? In this case, we have a reciprocal translocation where we have one segment of chromosome 9 moving over to chromosome 22 and vice versa. And when it does this, what we end up with is actually a case where this ABL gene, this CABL gene, and this BRCA gene come close to each other after translocation. And we end up with this altered chromosome. This Philadelphia chromosome is shown below. And what it does is when these two things come together and fuse on chromosome 22, on this shorter chromosome, so we have this section of the chromosome migrating over to this section of chromosome 22, so 9 to 22, we end up creating what's known as an oncogene, so a gene that can cause cancer. So it's really important, these genomic arrangements can be really important when we think about what is the genetic factor underlying something like cancer. Another example where we might take this approach is, say, looking at a comparison between human and chimp, where H represents the human genome and chimp is represented by the C genome. And what we can see here is by comparing these syntenic groups, these groups where we see all these patterns of the same genes, we can start to understand, what were some of the key differences between the genomes of the human and chimp genome. So, we see that chromosome 2, for instance, in humans is actually broken up into two different chromosomes in chimp. And we can identify regions of broad dissimilarity, say here on chromosome 9, or regions of high similarity here on chromosome 19. Like, we can find these various patterns of similarity and difference that can help us better understand whether the genetic reasons underlying the differences in phenotypes that we observe. So again, when we think about dot plots, we look for these general patterns. Basically, things that are along the diagonal here indicate synteny or similarity. Anything where we see like a break here indicates that there's some sort of syntenic discontinuity. And it's important that you know these types of figures, by the way, because we'll talk about them throughout the class. They'll pop up. But these syntenic discontinuities, like these are the areas of difference, right? The diagonal is the areas of similarity. We can see a line perpendicular to that diagonal. That indicates that one genome has inverted that sequence relative to another. And this is also an important thing to understand because that can lead to very different functional outcomes. So if I was looking at something like comparing turkey to chicken, right, one of the things I could see when I compare the dot plot of these two, like if I look at chromosome 2 in chicken, I can see that it seems to be made up of or broken up into two different chromosomes I would find in turkey, 3 and 6, right? I can also see that what I identified as chromosome 2 in turkey is actually chromosome 3 in chicken. So, this is one of the ways that we can go through and start to identify, what are the patterns associated with the evolution of the differences between these two domesticated species. And again, the data that is underlying that figure is actually, this sequence alignment data. So the question is, remember that I had like multiple options here over here on the right, like all these different options for sequences. How do I pick which one's the best? Well, generally I would use some sort of scoring system that would be based off of the matches. So the things that are aligned to each other in the same columns and the mismatches, as well as some penalties for putting in gaps. And, I could look at, there are a bunch of ways for me to calculate these, to score these things. There's Hamming distance, where I'm just looking at the numbers of mismatches. And so in this ATG, in this alignment here, right, I've got two differences, so that's a Hamming distance of 2. There's also a Levenshtein distance, and here it's a little more complicated because now I'm looking at the minimum number of edits that were required to change one string to another, and so that could include insertions and deletions. Now, not just mismatches or substitutions. So if I look at this alignment below, right, it would take one change here, one change here, and one change here in order to make these two sequences the same. So that's how I calculate the Levenshtein difference. When I do these alignments, there's also a way where I could either look at the whole sequences that I'm looking at, or I could just look at part of the sequences. And typically, what you would do in this process is try to figure out whether or not you can match everything, or you're just going to match segments of your genome that you can match, find your best match without any gaps. So you don't have to match the whole sequence. So global does all of it. Local is just looking for the parts that match the closest. So say I had a bunch of I could come up, if I had a sequence here, sort of based upon whether there's matching or mismatching, I go through and I could start to sort of score each of these matches and see how well I could score the system and align those two sequences. So there's a bunch of different tools that you could use, or algorithms you could use to do these different types of things. So on the left, this is a list of just a few alignment tools. There's like hundreds of these things. But those are some tools that we could use for global alignment. A lot of you guys have probably used BLAST. That's actually a local alignment tool, probably the most well-known one. But there's plenty of other types. One of them uses a very different methodology called a hidden Markov model approach. And that's the one that we use to actually identify protein family. And we use this based upon a pattern of conserved positional scoring matrices. So is the spot, as I move across a protein, do I find regions of the protein that are very similarly scored? And again, it's just an alignment method where I pick a scoring system. But this approach is one of the main ways that we, identify things that are structurally similar to each other, RNA and protein and things of that sort. So in the end, we're able to use all that genetic information, all this genetic approach, and all these multiple sequence alignments, looking at multiple genes, forming these multiple sequence alignments, looking at the patterns of similarity and the patterns of differences between these sequences to form a large entity like this tree that can help explain these evolutionary relationships. And after we've done all that, what we can end up seeing, as we can see in this case here in the African cichlids, is that anatomy and morphology is not always the best explanator. It is often not the best explanatory tool for looking at the evolutionary relationship between organisms. So this has been a sort of a brief synopsis of how we're going to be conceptualizing evolution in comparative genomics, where we're mostly looking at differences between species or comparative differences. It's not the only way that we're going to be looking at it, because we will talk about some other data types as we go through the class. But just generally understanding, like, this is the kind of broader context. And I think it's really important that you understand that how we're interpreting this data is always in the light and the context of their evolutionary history. Like, we can't really understand why something does the way it does unless we can account for its history, because all of these organisms are actually going to be biased in their function by what is their evolutionary history. And it's important that we take that into full consideration as we start to try to interpret it. So thanks for your attention, and look forward to the next topic of discussion.